Next function of membrane proteins is receptors for signal transductions. They serve as receptors for different molecules arriving from the bloodstream, for example, and acting on the cell. Those can be hormone molecules. Number 3. Attachment to cytoskeleton and extracellular matrix. Extracellular matrix is structures outside of the cell. Very often they just keep cells together. So proteins help to keep cells together. What about inner proteins? Inside there are structures of cytoskeleton, and those are fibers that keep organelles in place. So our membrane proteins help to keep our organelles, cell organelles in place. Next function would be enzymatic activity. So some of those proteins serve as an enzyme to speed up some chemical reaction. Number 5. Intercellular joining. So proteins help to join cells to the neighbor cells. And finally, number 6, cell-to-cell recognition. Cell-to-cell recognition, example of that can be when our white blood cell is floating through the blood and finds some other cell, and our white blood cell needs to know if it's a self cell or it's a foreign cell like bacteria. So by checking those proteins, special proteins, white blood cells can recognize our self cells. Membrane transport. That is transportation of substances through the cell membrane. Our plasma membranes are selectively permeable. It means that some of the molecules can easily pass through the membrane. Those are usually tiny molecules. Other molecules cannot do that. In most of cases, those are larger molecules, so selectively permeable. There are two types of membrane transport. Passive processes do not use any ATP molecules for energy. Substances move through the membrane down its concentration gradient. What does that mean? Let's say a concentration of some substance is higher on one side of the membrane and lower on another side of the membrane. In this case, substances will be moving across the cell membrane from the area of high concentration to the area of lower concentration. Now, active processes require ATP energy, and it only happens in living cell membranes. Okay, we're starting from passive processes. There are 4 of them that you need to know. Simple diffusion, carrier-mediated facilitated diffusion, channel-mediated facilitated diffusion, and osmosis. Let's make it clear first. So there are 2 types first, diffusion and osmosis. Diffusion can be simple and facilitated. And finally, facilitated diffusion can be carrier-mediated and channel-mediated diffusion. We're starting from number 1, simple diffusion. That is a cell membrane. There are a lot of substances on one side of that membrane, and those substances are small enough they can cross right between those phospholipid molecules. As I told you before, when concentration is higher on one side and lower or non-existent on another side, substances start crossing the membrane from the area of high concentration to the area of lower concentration until concentrations equalize on both sides of the membrane. So it's a simple crossing through membrane from the area of high concentration to the area of low concentration. Next, on facilitated diffusion. Pretty much the same thing. Substances are crossing from high to low concentration, but now they are not going through the phospholipid bilayer, they are going through the special proteins. That is facilitated diffusion, going through the proteins. Those proteins, there are two types of them, carrier proteins or channel proteins, and both of them exhibit specificity or selectivity. It means they only allow certain molecules to go through them, not all of the molecules. Also, they are saturable. If we have a lot of substances that want to go from one side of the membrane onto another side, but we don't have enough of those proteins, it slows down the diffusion because proteins are saturated. And finally, those proteins can be regulated in terms of activity and quantity. It means our body can stop them from crossing, from letting those substances to cross, and our cell can regulate amount of those proteins. So one type of proteins is called carrier protein. It means those proteins are usually open and substances just cross through the channels inside of them. Next one. There are two types of those channel proteins. Leakage channels are always open, and gated channels can be sometimes open, sometimes closed. They are controlled by chemical or electrical signals. It means normally that gated protein is closed right here, as if it has a door right here. It would be a piece of protein covering it. And when some certain chemical signal or electrical signal arrives, the protein opens and allows the substances to cross from area of higher concentration to lower concentration. Next one, osmosis. Osmosis is a movement of water across a selectively permeable membrane. So, when water is going through the membrane, it can go straight through the bilayer of phospholipids right here, or it can go through the special proteins for water, and those are called aquaporins. Please memorize that name. So, aquaporins, proteins for water. And again, water is moving from the area of high concentration of water to the lower concentration of water. Let's talk more about osmosis. That is a cup which is divided by selectively permeable membrane, and concentration on one side is much higher than on another side. Those molecules are too large and they cannot cross the membrane, but water molecules can. Where concentration of water is higher, on the left or on the right? Again, I'm not asking about those molecules of some substance. I am asking about water. You can see that there is more water on the left side. So, because of osmosis, water starts crossing and moving to this right portion of the cup, from the area of high concentration of water to the area of low concentration of water, until concentrations equalize. That's what happens. And water can enter or leave the cell, and amount of water is important because it changes cell volume. And if we change that cell volume a lot, it can disrupt cell functions, or it even can kill the cell. Okay, let's try to solve that problem. There are two solutions.